Okay, so for question number 1, we're given this RNA sequence, and I abbreviated a little bit since we were given it. And part A is asking to find what end is the 3' end and which end is the 5' end. And it tells us that it's produced from right to left. So that shows us that the 5' end is going to be over here on the right hand side, and the 3' end is going to be over on the left hand side. Part B is asking what the template strand of DNA is going to be that would have coded this strand of RNA. And so if we look, we can match them up, and that would give us this sequence right here. And this is the template strand of DNA. And then for part C, we're looking for the coding strand, which is going to be the strand on the opposite side of the template strand. And so those match up as well. The sequences match up right here. And that's how we get our coding strand. And then for part D, we're asked to draw a picture, and this is our picture of this taking place. And so we have the 2 DNA sequences right here. We have the coding strand and the template strand. This is 3' end for the coding strand, 5' for the template strand. They're going anti-parallel, obviously. This is our RNA polymerase II, this big blob right here, and this is traveling this way, so it's going to be going to my right here. So our TATA box, our TATA box is right here, and that helps to initiate it, so it's going to be behind which direction the RNA polymerase is traveling. And then here's our mRNA being produced from 5' to 3' here from our RNA polymerase II.
Go.
So for question 2, we have a bunch of fill in the blanks. So for A, we got the TATA binding protein, which is the TBP, binds directly to the TATA box, while helix turn, helix motif binds directly to the -10 and -35 sequences. For B, we got guanylyl transferase is a eukaryotic enzyme that links the 5' carbon of one nucleotide to the 5' carbon of another nucleotide. And then for C, we got group 1 introns use the nucleoside guanosine as a cofactor during catalysis, while group 2 introns use adenine as a cofactor during catalysis. For D, we got the CPSF, which is the cleavage and polyadenylation specificity factor, is an endonuclease that assists in terminating transcription in eukaryotes. For E, it says, in lesson 9 we learned about the general transcription factors, and TFIIB is a GTF, which acts as a bridge subunit to link TFIID to TFIIF RNA polymerase II, whereas TFIIH is a GTF, which acts to phosphorylate and activate RNA polymerase II. And then for F, we got the spliceosome is composed of subunits called SNRPs. These spliceosome subunits are composed of 2 components, proteins and small nuclear RNAs.
Go.
So for question 3, part A, it says indicate 2 proteins in either prokaryotes or eukaryotes that act as a helicase to separate 2 DNA strands during transcription. And so we got TFIIH and RNA polymerase II. For part B, it asks to indicate 1 protein in prokaryotes that acts as a helicase to separate RNA from DNA during transcription, which is the Rho protein. For C, it asks for 3 molecules in eukaryotes that function during RNA modification as ribozymes, which we got RNA 5' triphosphate, guanylyl transferase, and methyltransferase. And for part D, it asks for 1 protein in prokaryotes that acts as a transcription factor, and we got the sigma factor. And for part E, it asks for 1 enzyme in eukaryotes that synthesizes a long string of identical RNA nucleotides, but does not use a DNA strand for RNA synthesis. And for that, we got RNA transcriptase.
Okay, so for number 4, she kind of explains in the video, Sister Mayo explains in the video more about these. So I've color-coded these for, the green is going to be mRNA, blue is going to be your DNA template, and then pink is going to be coding. So in the problem it says that for your favorite gene, it contains 4 introns. So for B, it says that you have a mature mRNA, which means that splicing has already taken place with the introns. And so it just wants to see, we're trying to see what it's going to look like. So you have your mRNA strand, and it's going to be binding to the DNA strand. But since you have those 4 introns, and those were taken out, that DNA sequence is going to be gone, so it just can't bind to anything, so you're going to get this loop. And so you're going to get 4 loops because of the 4 introns, or those 4 sections that aren't in the mRNA, so the DNA can't bind to it. Then when it starts coding, the DNA coding, if there's no mRNA binding to the DNA, then it's going to be able to bind with it. But since mRNA is stronger, once it's binding with it, the coding won't have anywhere to bind to, so you're going to get these R loops. But then, since mRNA isn't binding with these intron loops, then the coding will start binding here. And so you see these 4 intron loops, and then you see the 5 R loops happening. So then with part C, it says that there's an issue with one of the introns, or it's defective, and so it's not going to end up splicing out in the mRNA. So you're really only going to have 3 areas that were spliced out because of those introns. And so same thing, you're going to get loops, but you're only going to get 3 of them this time, since one of those introns is defective. And so I just put like a little X to mark where it was, because I think it said it started on the 5', the 5' most intron. So since this is my 5', and I just took out that one right there. And then for D, it says that your 2nd and 4th intron are defective. And so in this one, again, I just put X's where that intron isn't going to be, because it didn't splice out, and so it is actually going to be able to bind there. And so you'll only get 2 intron loops here, and then you'll have 3 R loops. So then I'm actually going to go back to part A now. This just made more sense to me this way. And so it says that the pre-mRNA for your favorite gene is hybridized to the genomic DNA copy. So when you have the pre-mRNA, splicing hasn't occurred yet, so you're going to have the whole DNA sequence there still. And so when the DNA is going to bind to that whole thing, you're not going to get any intron loops yet, since it's the pre-mRNA. So then when the coding happens, it'll bind when it's not binding with mRNA, but then you'll get this big loop here, since it can't bind with anything in that area. And then once it's to the end of the pre-mRNA, then it'll start binding to the DNA again.
Perfect. Okay, so for question number 5, we have a list of all these things, and we're trying to figure out which ones go in these various columns. So for part A, we're trying to figure out which one of these go into or are found in prokaryotes. So we put the -35 sequence, RUT, and NUSA are all things that are found in a prokaryote, specifically E. coli. And then part B, we're looking at a eukaryote. So we have -25 enhancers and silencers. They kind of are together in there. Group 1 introns, the GC box, TFIID, and SNRNP are all things that are found in the eukaryotes. And then for part C, we're looking for which ones are found in initiation. And obviously -35, -25, those are those start initiation, GC box right here, and then TFIID. And then for part B or part D, we're looking at termination, so obviously the RUT and the NUSA. And then for part E, we're looking which one of these or which ones of these are DNA sequences. And so -35, -25, the enhancers and silencers, these are all found in DNA, and the group 1 introns are a section of DNA, and the GC box are all found in are found compound sequences of DNA.